Yes, we're good. Thank you, Helen. Over to you.
God, I hate that. Okay, thank you for your patience. So today I thought, because it's a developmental audience, I'd actually tell a bit of a historical story about how a project we've been following for quite some years, and it gives me a chance, because it's a developmental audience, to actually talk about some neural tube stuff. So as many of you know, we have had a long-standing interest in this receptor called Neogenin, which we cloned many, many years ago because it was, and discovered that it was very, very closely related to the netrin receptor DCC. Since then, there has been a lot of work done on Neogenin, including from our own lab, and it turns out that Neogenin's got a much broader role than DCC, and one of the things that fascinated us was that it's always expressed in stem cell populations. And so work from many labs has now shown that it's involved in things like angiogenesis, bone formation, and also is involved in a disease called hemochromatosis, and it's involved in iron hemostasis. It is clearly an axon guidance receptor, and it's since been identified as two distinct ligands, netrin and RGMAs, which is different to DCCs. And we know that when it interacts with RGMA, it causes axon repulsion. And we know when it interacts with netrins, it causes axon attraction. We know that these processes work via actin, and so RGMA triggers actin depolymerization, whereas netrin triggers actin polymerization, but we really don't know much about it in terms of how those different mechanisms occur. So our labs over the years have spent a lot of time working on Neogenin, and we've got the new evidence that it controls dendritic spine formation, and we've previously published showing it's involved in adult neurogenesis, which is where it controls stem cell cycle exit, differentiation, and migration. But one of the things that really fascinated us was it is expressed highly in the neocortex here during cortical development, and all other areas along the developing CNS in the areas where the radial glial stem cells reside. So we decided, rather than take a complicated approach and look at mice, we started off by looking in zebrafish. Sorry, we did do zebrafish, but this is our Xenopus study, where we used an old-fashioned approach, which are called morpholinos, which allowed us to specifically knock down Neogenin in Xenopus embryos. So very similar to shRNAs, but they inhibit translation. And so when we introduced Neogenin-specific morpholinos into two-cell stage embryos of Xenopus, what we found was that we were getting neural tube closure defects. So in the Xenopus around stage 13, there's no neural tissue, but over the next few stages, what happens is that the neural tube develops. And here in the control morpholino animals, you can see you've got closure of neural tube, while when we knocked down the Neogenin, the neural tube remained open. And you can see here when the quantification, we had quite significant delays in neural tube closure. So the neural tube starts off as a plate of neuroectoderm. And over that time period, the neuroectoderm sinks, and these neural folds start to appear, and they continue to rise up, and then as the neural tube continues to form, that you get bending here, and finally it fuses at the midline here. So this process involves quite a lot of complicated developmental events, but basically for this talk, all you need to know is that the neural tube starts off as a neuroectodermal cell type, and then through several processes, including the induction of apical basal polarity and cell-cell adhesion, it converts to a pseudostratified neuroepithelium. So neural tube closure, you have a neuroepithelium, and that neuroepithelium then gives rise to the radial glia, and therefore all the neurons in the CNS. So when we had a look at where Neogenin was expressed, here we're looking at an anterior view of the, or dorsal view of the developing Xenopus neural tube. So initially at stage 13, it's flat, and as the neural tube, neural folds rise and then fuse. You can see here, this is an in situ of neogenin, and you can see it's clearly present in those neural folds all the way through that process. And this is just a cartoon showing that neogenin is always in these neural folds, and its ligand RGMA is always at the midline. So it's always forming a gradient out towards where neogenin is expressed. So when we look more carefully at these embryos, what we saw was a severe defect in the neuroepithelium morphology and function. So this is the control. You can see here, like going down here, and this is more magnified here, the beautiful pseudostratified neuroepithelium of the neural tube here. In the neogenin morphins, we see a lot of cell death, and for those cells that are remaining, severe disruption of the neuroepithelium, the cells are rounded, and you see a lot of de-adhesion. And when you knock down RGMA, although the phenotype is not severe, you still get the same phenotype. So we see completely a disruption of the neuroepithelium and lots of adhesion defects. So just to talk about the adhesion story a bit. So it's the neuroepithelium. It has the apical membrane faces the lumen, and the basal processes are on the outer side of the neural tube there. And so these neuroepithelial cells are highly polarized, and the adhesion at the apical surface is maintained by cadherin-mediated adherence junctions. And the cadherin-mediated adherence junctions is also reliant on the actin cytoskeleton. And so when we knock out neogenin or RGMA, we're seeing the collapse of these junctions, and we presume, and also therefore the loss of the pseudostratified neuroepithelium. So going back to the mouse, having seen this in Xenopus, we decided that we should probably go have a look at what's happening in the cortex of the mouse and during cortical development. So this is just to remind you a slight story about cortical development. Here we've got the neuroepithelium of the neural tube. These cells divide symmetrically to reproduce and propagate the stem cell pool, but eventually as corticogenesis initiates, these cells start to divide asymmetrically, producing one new radial glial cell and a neuron. So this highly polarized epithelial structure of the radial glial cells is important because it controls key facets of cortical development. It determines whether the cell has an important role in determining whether the cells divide symmetrically or asymmetrically. It controls the actual cytoarchitecture of the cortex, because the newborn neurons, which are born down on the apical surface, use these basal processes to migrate to their appropriate layer. So as in the neural tube, these at the apical membrane of these radial glial cells, which are derived from the neuroepithelium, they are maintained in a cohesive tissue via the cell adhesion between cells, which again is cadherin mediated, and these are adherence junctions. We know that when you disrupt these junctions, you completely disrupt the cytoarchitecture of the cortex, because the new neurons cannot migrate, and you end up with a range of cortical malformations, such as these heterotopias here, where in this case, you have a second layer of neurons below the cortical plate, and here you have neurons protruding into the lumen of the ventricles, which these sort of heterotopias have severe effects such as epilepsy and intellectual disability. So we wondered whether neogenin had a role in this process, so we undertook some in utero electroporation experiments where we used an shRNA approach and co-transfected GFP at embryonic day 12, and you can see that GFP is highlighting the beautifully polarized radial glial cells here, and also some of the new neurons that are born over this period when we section the animals at E14. And so when we had a look at these this cortex during development after knocking down neogenin, here, first of all, you can see that We have a beautifully formed cortex when we use the control siRNA. Here are the radial progenitors all polarized, the newborn neurons moving up into the cortical plate. When we knock out Neogenin, we see complete disruption of the ventricular zone. A lot of the progenitors have actually migrated up into the cortical plate, and those that are remaining have lost their polarity. And you can also see defects in neuronal migration, because we see protrusion of neurons into the ventricle, and also inappropriate layering of neurons in the ventricular zone. So it's very similar to the cortical heterotopias that are seen when you disrupt neuronal migration. So we also had a look at what was actually going on in terms of junction. So here we're close up of the apical surface, and we're using beta-catenin as a marker for adherence junctions here, and you can see they're tightly aligned at the apical membrane, and this is just a high power image, and you can see the beta-catenin highlighting the junctions between the different radial glial cells. So in the Neogenin knockouts, what we actually see is the loss of all these junctions here. Again, the radial glia are rounded and no longer highly polarized, and actually the loss of beta-catenin here indicates loss of junctions. And so as we saw in the neural tube, we're seeing a defect in adhesion and presumably defect in adherence junction formation. So what we're showing here is that if you remove Neogenin, you lose the adhesion. That results in defects in polarization and that leading to cortical malformations. I don't have time to really go into it at the moment, but the radial glia eventually around birth convert to ependymal cells, which line the ventricles, and they are also the adhesion of the ependymal cells is also dependent on the same type of cadherin-based junctions. And in Neogenin knockout mice that we see severe hydrocephalus due to the loss of these ependymal cells. And a lot of data we have, which I'm not showing you, is again showing that if you remove Neogenin, you lose the adhesion due to loss of cadherin-mediated adhesion at these junctions. So we wondered what was going on here. And so what we did was turn to a model, an epithelial model. So a radial glial cell is an epithelial cell, it's just a different shape. But we could do a little bit more of an in-depth analysis of what's happening when we knock out Neogenin. So we use these cells. We basically used the shRNA approach, and we looked at junction formation. So these cells can be grown in gels as cysts, and they form little neural tubes, essentially. So here you can see, here's a tube of cells. They're apically polarized with apical surface at the lumen and basal surface on the outside, just like a neural tube. And when you look at them in 3 dimensions here, the APKC is a marker for the apical membrane. You can see the cells closely associated with DAPI, and the cadherin here is showing the formation of cadherin-based junctions within these cells. So when we knock out Neogenin or the ligand RGMA, we completely lose the structure of these beautiful little cysts. And but with the Neogenin knockout, it's again much more severe, but you see the same phenotype in both. The cells are rounded, the cells, you can't clearly see a lumen because the apicobasal polarity is disrupted, and so is, when quantifying the number of actual adherence junctions, we see a significant decrease in junctional formation, as you might predict. And this is sort of a confirmation of what we were seeing in the neural tube. So when we had a closer look then using monolayers of these cells. So these are monolayers now. You can see Neogenin at the junctions clearly, which are outlined here by cadherin. And when you knock out Neogenin in these cells and you look at them at the membrane level, you can see the severe defect. And so when we also stained these cells with just the membrane-bound form of GFP, so we could look a bit closer. And what you can see here is the cells are just jiggling around because the green cells, the green lines are the junctions, and because they're under tension, you can always see a bit of movement. But when you looked at these knockout cells, you can see that the membranes are blebbing, they're doing quite weird things, and they're very dynamic. So this suggests that, in fact, what's going on here is that the membranes are not just de-adhering, but the tension that the structure, the actin structure and the other molecules that maintain the tension are not doing their job in these cells. So we had a look at the actin cytoskeleton, and as I said before, these cadherin-mediated junctions require the interaction between cadherins and actin cytoskeleton below the junctional membrane. So here you can see staining with phalloidin, and you can see that the actin is F-actin is beautifully outlining the junctions here. And look, when you look at neogenin knockdowns, you can see that the junctions in these cells disappeared, but moreover that there's complete disruption of the actin cytoskeleton. We then looked at whether whether it was just that the cytoskeleton fell apart, or whether there was a defect in actual formation of the skeleton, in that polymerization of actin. And so we looked at the incorporation of G-actin at these membranes in the knockouts and wild type cells, and you can see you can see by the decrease in the amount of G-actin fluorescence at each of these junctions that what we're actually seeing is a failure to actually polymerize the actin. So another thing you need to know about these cadherin junctions is that it's not just normal F-actin that's important in maintaining the cadherin adhesion, but it's branched actin, where you have a filamentous actin with branches jutting off at 70-degree angles. And this branched actin is produced by a specific enzymic complex called ARP23, which actually initiates a branch off the mother actin. But this ARP23 is inactive, and it requires this huge complex, which is RAC activated, called the wave regulatory complex, to activate the ARP23 itself. And so if you remove the wave complex, then the junctions fall apart. So one thing when we were doing these experiments, which was not known, was, okay, if you need ARP23 and the wave complex, how do they get recruited to the cytoplasmic domains of the cadherins? Because that was not known. And also, while we were doing these experiments, a paper came out by Chen et al. That had identified this sequence in proteins that interact with actin, called the wave interacting receptor sequence. Very awkward name, but this sequence on the cytoplasmic domains is a docking site for the wave complex. And so we found that Neogenin had one of these particular docking sites. So we decided to see whether this was what was happening in our animal models. And so what you can see down here is it's a very small 6 or 7 amino acid motif, with these 2 purple amino acids being key to the docking site. And so our question was, does Neogenin anchor in the wave complex and ARP23 to cadherins? So we went back to our cell model, monolayer cell model, and we did the same neo-siRNA knockdown study, but then tried to rescue with wild type, or with a Neogenin in which we mutated the docking site. And here what you can see in this top graph is that when you knock out, what we're doing is measuring the fluorescence across the junction here. So this is E-cadherin fluorescence at the peak, which tells us whether the junctions are formed or not. And when we use the siRNA, and you can see a significant decrease in junction formation, that's completely rescued by the wild type. But when we tried to rescue with the mutant, we don't get any rescue, suggesting that in fact Neogenin is having its effect on junctional formation via its ability to recruit the wave and ARP complexes. So with that information, we went back to the mouse and we electroporated. We took a dominant negative approach here. We've also done this in a knockout way, so I don't have time to show you this data, but what we did was just take the cytoplasmic domains of wild type and the mutant Neogenin and put a myristylated groups on it so that we could make sure it went to the membrane. And then we used in utero electroporation into our E12 embryos and then looked at E14 to see what was happening when we disrupted the interaction between Neogenin and the wave complex. So this is the mutant here, and we did a lot of experiments. So we know that this mutant does not have an effect on the effect of Neogenin interacting with the wave complex, as you would expect, because it cannot dock with it. It's not having an effect because it can't interact with the wave complex. And when we electroporated this into the embryonic cortex, here you can see a beautiful cortex. PAX6 is labeling the nuclei of the radial progenitor cells. Here you can see the junctions down at the apical surface as indicated by NK adherence staining, and this is just a high magnification. When we electroporated this wild type version of the cytoplasmic domain, we got a severe phenotype. Now, this cytoplasmic domain with the wild type sequence is acting as a dominant negative, and we have shown that when this actually interferes directly with the interaction between Neogenin and the wave complex. So what we see here is, as we did when we did just straight knockouts of Neogenin, complete disruption of the ventricular zone, and here you can see complete loss of adherence junctions indicated by the loss of NCAD staining. We also looked at F and G actin, and again, when we use the mutant, as would be predicted, we see normal F actin accumulation at the junctions, whereas in dominant negative cortexes, we've lost the F actin and we're starting to see accumulation of G actin, indicating loss of polymerization. And just to confirm that it's the wave complex that we're interrupting, here again with the mutant version of dominant negative neo, you can see that we've got wave and ART23 in these junctions here, but with a dominant negative prevents the interaction between wave and Neogenin, and the result of that is that we lose wave and ART2 from the junctions. So
sorry to interrupt, Helen. 3 minutes left. Thank you very much.
Yeah, that's right. No, I'm pretty close to finishing now. So this is the summary slide here. So what we're up to, what we've now shown is that over the many years we've been looking at this, is that Neogenin controls the adhesion of radial progenitors and neuroepithelial cells by controlling branched actin polymerization. And so we've convincingly shown that Neogenin anchors the wave complex and ART23 to the coherence. This promotes the polymerization of branched actin, and this thereby stabilizes the adherence junctions and maintains tissue integrity. And then when that interaction is disrupted, it has quite a lot of serious consequences for the embryo and the neonate, depending on what stage you're looking at. So we see heterotopious disruption of cortical architecture in the developing cortex. Postnatally, we can see hydrocephalus, and this also now explains our very early observations of neural tube defects. And so the last slide, I just wanted to go back and say Neogenin's probably most famous for its ability to guide axons. We know that axons navigate through filopodia and lamellipodia, and lamellipodia are strictly dependent on the wave complex and ART23 for their movement forward, and so we suspect that that's how Neogenin is having its effect. And in our new data, where we show that Neogenin and its interaction with wave is, we've shown that Neogenin is important for spine formation, and we've now shown that it requires a wave complex. So the ability to control the actin cytoskeleton has quite a profound effect across development in terms of in terms of the morphology and cytoarchitecture of the embryo, and also it is now emerging that it has important roles in circuitry formation postnatally. So I'll leave it there and just highlight Natalie, Connor and Amanda, who did most of this work. Thank you.